Stem cells are pluripotent and specialized cells characterized by their long-term self-renewal and their ability to give rise to differentiated cells from the three germ layers under certain conditions. Due to these properties, stem cells have a great potential in regenerative medicine, disease modeling, and animal engineering. For years, embryonic stem cell lines destined to research purposes have been mainly derived from mouse embryos, but in general, the derivation efficiencies achieved have been low. In this video, we show a simple protocol to derive mouse embryonic stem cell lines using standard conditions that provides reasonably high derivation efficiencies comparable to the efficiencies obtained when using modulators of signaling pathways such as 2I.
In this protocol, we'll use human foreskin fibroblasts as feeder cells for ESC derivation and culture. Before starting the ESC derivation process, it's necessary to inactivate the feeder cells. First, take a cryogenic vial of human foreskin fibroblasts stored in liquid nitrogen and thaw it by immersing the vial in a water bath at 37 degrees C. Once thawed, dilute the content in DMEM supplemented with fetal bovine serum. Seed the dilution in a Falcon flask and culture it until reaching the confluence stage. Next, to inactivate the fibroblasts, dilute mitomycin C in DMEM supplemented with fetal bovine serum and incubate the cells for 3 hours.